There are different kinds of phylogenetic tree reconstruction methods, and mainly they have been divided into two types. One of them is character or sequence-based methods, where we look into individual characters independently, or we look into the sequences. Whereas here in distance-based methods, we take those sequences and we compare them with one another, and we come up with some distance matrices. So character-based methods, they assume that each character substitution is independent of its neighbors. And in this character or sequence-based methods, again, there are two major types of the methods. One of them is maximum parsimony, and the other one is maximum likelihood. Maximum parsimony means a parsimonious tree where you can do some parsimony, bachat. So in this case, you built a tree with fewest possible changes. So minimum changes you should make in order to make that tree, and in this method the minimum changes are the good ones. Whereas in the maximum likelihood approach, basic idea is to build a tree based upon some mathematical models. It performs analysis on each position of the alignment, and using a tree model for nucleotide substitutions, it will try to find the most likely tree out of all trees given in a data set. Distance-based methods, they take the sequences and then they come up with their pairwise differences and or distances, and then we arrange these distances in a matrix. We call it as distance matrix, and then the tree is made while using that distance matrix. Again, there are two major types. One of them is UPGMA, unweighted pair group method with arithmetic mean, and the other one is NJ or neighbor joining method. In UPGMA, it is a simplest method for a reconstruction of phylogenetic trees. So what we do here, we assume molecular clock hypothesis, where we believe that there is a constant rate of evolution between different organisms or sequences for which we are making those trees. So we believe these different organisms or units, they change or evolve at a constant rate. So we have same rate for everybody. So in this way, the trees lengths of different branches is approximately same for all of them. Whereas in neighbor joining, we do not assume this molecular clock hypothesis. It allows for unequal rate of evolution in different branches of the tree. Here is in this chart or in this flow diagram that is present in your book mount, we can put all these methods all together, and we can come up with a strategy on what to do if we have some sequences. So we choose a set of related sequences, and in the next step what we do over here is in number 2, we obtain their multiple sequence alignment, and then we see if we find some strong sequence similarity. If the sequence similarity is high, we can do maximum parsimony method. If it is not high, then we can look into some low similarities, but still we can recognize some sequence similarities as we see in step 4. So in that case, we will use the distance methods. If we are unable to get those even some similarity, then our choice may be maximum likelihood methods. And then we analyze our results and we see whether our data supports the predictions or it does not. So this is the overall strategy for tree reconstruction, starting with any set of sequences. So we see in this section that there are different types or methods of phylogenetic tree reconstructions. There are distance-based methods that depends upon the distances among the sequences, whereas we have characters or sequence-based methods where we assume that each character substitution is independent of its neighbors or if independent of other nucleotides.